Hello everyone, thank you for your patience. We're now ready to begin this month's Glural-Siler seminar series. My name is Tim Davis and I'm very pleased to be hosting Dr. Silvia Newell. Dr. Newell did her undergraduate work at Smith College in Massachusetts. She then went on to do her Ph.D. at Princeton University, followed by a postdoc at Boston University, and has been a faculty member at Wright State University since 2014. I've worked with Dr. Newell in the past, and she does some very good science in multiple different systems, and today her talk will be focused on nitrogen cycling in eutrophic systems with a focus on Lake Taihu and Lake Erie. So with that, I will let Silvia come up and begin her presentation. For those of you who are on the webinar, please feel free to chat your questions that you might have to the host of the webinar. At the end of the presentation, we'll then read off any questions that come through the webinar, and Dr. Newell will then answer those questions through the mic, so you'll have a way to interact through the chat box. So with that, Dr. Newell.
Thank you for that nice introduction, Tim. So today I'm going to be talking about nitrogen cycling in two of the systems that we're currently working in, like Tim said, both of which are very eutrophic. So one of the reasons I'm so passionate about the nitrogen cycle specifically is that it's one of the most altered biogeochemical cycles that we have. This graph here from Galloway et al., if you look at the purple line, this is our population growing, and you can see that although biological nitrogen fixation has increased a little bit, mostly we've got a huge amount of total reactive nitrogen coming up, both from the implementation of the Haber-Bosch process and additions of fertilizer, also fossil fuels, etc. And these numbers are a little bit old. We're now up to well over 200 teragrams of nitrogen per year, which is about 4 times the 50 teragrams per year they get naturally fixed biologically. So we are drastically altering the nitrogen cycle. And this increase in not just nitrogen but also phosphorus nutrient loading can have really significant impacts, as I'm sure you all know. Got harmful algal blooms. This nice green scum here is a picture that my Ph.D. student Daniel took last year when he was out with NOAA Glural in the western basin of Lake Erie. Got toxin production, oxygen depletion, especially in the Gulf of Mexico, fish kills, etc. But when we talk about these problems and when we talk about them from a policy perspective, a lot of times we're really only talking about changing the amount of phosphorus loading and not the amount of nitrogen loading. And I, along with everybody else I know, thinks that phosphorus is really important, but when it comes to talking about policy, I think we need to consider both, and I'm going to give you my argument for why. But for the moment, I'd like to point out this very nice paper by Silvia et al. Discussing the assessing and addressing the re-eutrophication of Lake Erie Central Basin hypoxia. And they talk about phosphorus about 150 times, and I think nitrogen is mentioned once on the y-axis of a graph pretty far into the paper. This is one of my favorite slides to make this point. It's from Hans Perl. It's from a series of mesocosms that they did in Taihu in October of 2008. And if you look at the filters from these mesocosms, you see that in the control where they've added no nutrients, it's still kind of green in October. There's still some Microcystis there. And when you add phosphorus, it gets a lot greener. You know, you've definitely stimulated a bloom there. And when you add nitrogen, it's even greener. And when you add nitrogen and phosphorus together, it's so dark, it's almost black, even on my screen with its better color. So I think this is a really easy way to look at this and say, okay, well, maybe, and especially in Taihu, this changes. Which nutrient is limiting? And to me, the point is not so much which nutrient is limiting at any given moment in time, but what's the importance of both things over the year annually within an ecosystem. And I like to think about the changes that doctors and other people have given regarding diet over the last 30 years. For a while there, you weren't supposed to eat fat. You weren't supposed to eat butter. You were supposed to have margarine. And then they discovered your body can basically never break it down again and pretty much stuck with it for the rest of your life. So maybe not such a good idea to have fake fat. And then it was fake sugar, and you weren't supposed to have, sugar. You were supposed to have everything low sugar. And they put in things like aspartame and whatever, which turns out not to be that great for you either. And now it's carbs. You're not supposed to eat carbs. Carbs are the devil. Please don't eat them. They're very bad. Everybody go paleo, etc. And I just think that saying whether we should control nitrogen or phosphorus is like saying, which things we should eat. And I'm very much in the Michael Pollan camp of, eat food, not too much, mostly plants. Well, I think a balanced diet is important for pretty much everything that lives and not just people. And there's a lot of data to back this up. This is an earlier version of a graph that has recently been published in the Pearl et al. ES&T paper that came out a couple months ago that I'm on, and this is from our co-author Wayne Wurtzbach. And it's looking at whole lake nutrient additions where you look at either just nitrogen additions, just phosphorus additions, or nitrogen and phosphorus together. And you can see that when you add phosphorus, quite a bit of the time you get an increase in chlorophyll, our proxy for biomass. When you add nitrogen, there's often a very big increase in chlorophyll. And when you add nitrogen and phosphorus together, a lot of the times it's way up here off the scale. I mean, to me this says, hey, we're in this situation already. We have already hit we've added too much nitrogen and phosphorus in most of our eutrophic lakes. And the question is, how do we back it off from there? Heck. And if you want to go backwards, and I don't really think it works this way, if you reduce phosphorus, yeah, you're going to get a lower amount, but there's still going to be a ton of extra chlorophyll essentially from baseline. If you add or reduce nitrogen, you're going to get a lot too, but it's still not where you want it to be. I really think we have to control both things together. But one thing to notice about this slide and many other slides is that the nitrogen that was added in all of these additions is nitrate. And nitrate is only one of many forms of available nitrogen, and it is oxidized. It's a lot more difficult to use. And what we've been seeing is an increase in reduced nitrogen as fertilizer has shifted toward global urea additions in the last 20 years or so. And we see this change reflected in the kind of blooms that we see in the western basin of Lake Erie. From the 1960s and 70s, we saw a lot more Anabana, I refuse to do Dolichospermum, than Aphthana zamenon, versus what we're seeing now, which is a lot of Microcystis. And the main difference between these two things is that Microcystis cannot fix nitrogen. And nitrogen is very difficult to fix. It's got a triple bond, it's hard to break, requires a lot of ATP and electrons, and there's not a lot of point to do it if you don't really need nitrogen. And yes, there are parts later on in some blooms where you draw all the nitrogen down and it becomes competitive to actually fix nitrogen, but a lot of the time it's just not useful really to fix nitrogen when there's so much available ammonium being recycled. Which brings me to reduced nitrogen. The form I'm going to focus on the most right now is ammonium. I also think that urea and the dissolved organic nitrogen are important, and we're starting to work on those, but ammonium is pretty straightforward to look at. Now, things like diatoms can use and store nitrate efficiently, but most phytoplankton, including Microcystis, especially Cyanobacteria, greatly prefer ammonium and other reduced forms of nitrogen. We tend to ignore ammonium. We tend to think it's not that important because it's not the main thing that is measurable. But I strongly remember my Ph.D. advisor, Bess Ward, giving this talk in the middle of biological oceanography, and it wasn't necessarily about nitrogen, but she said, imagine a frat going to buy supplies for their party on a Thursday. And let's say they buy a bunch of veggie trays and they buy a bunch of beer. And then they have some raging keggers for the next 2 days. If you go into their fridge on Sunday morning, what are you going to measure? I mean, what are you going to see? Are you going to see a fridge full of beer still, or are you going to see all the broccoli and the rest of the, like vegetable leftovers there? Well, I mean, I think the answer is pretty clear. It's the thing that is most desirable that's getting used up the fastest. And so just looking at concentration is not sufficiently representative of what's really important. And so when you actually look at what it takes to assimilate various kinds of nitrogen in phytoplankton, got a nice diagram here from Beversdorf et al. 2015 and the Glibert et al. 2015. And the main point I want to make in these very complicated slides is just that if you take up nitrate or any other form of nitrogen, including urea, everything has to get reduced to ammonium to be able to be incorporated into amino acids. We've also seen a lot of new work lately showing the importance between nitrogen and toxicity in Microcystis and other species. These toxins are very nitrogen heavy for the most part. It takes a lot of nitrogen to make them. We don't really know why they make the toxin yet. Tim Davis has a lot of theories, heard some other theories from Wayne Carmichael, but the main point is that the availability of nitrogen is probably what is limiting the production of toxicity. And Tim has published some work showing that reduced N-form additions to non-N-fixing cyanobacteria can increase toxicity. Kuniyoshi is showing that low ammonium concentrations can inhibit the production of toxin. And then this Harkey et al. Showing that ammonium and urea uptake may lead to both increased Microcystis biomass and toxin production. And this is that graph over here from Harkey et al. And you can see that the Microcystis biomass up here is the greatest when you're adding ammonium or urea, at least when you're adding only phosphorus. And down here is the microcystin, and you can see that when you add urea specifically, there was a giant increase in microcystin production. But it's not just toxicity, and it may not be, and it's definitely not the only driver of biomass, but what form of nitrogen is there does strongly impact the phytoplankton community structure. And that is so important because diatom blooms are much better to have for an ecosystem, not only because there's more biomass per cell, so you get clear water, they don't make toxins, they have better DHA and other fatty acids, they're better food for up the food chain, but they don't form surface scums. And cyanobacteria, unfortunately, are the opposite on all of those things. And if you look at this graph here from McCarthy et al. 2009, this is a long-term data set from Okeechobee, and you can see that as you increase the ratio of reduced to oxidized nitrogen, you see a shift away from diatom blooms and a shift toward cyanobacterial blooms. Cyanobacteria are very competitive for ammonium and other reduced nitrogen forms, and when those become more available, cyanobacteria become more dominant. And I really like this graph here that Gobler and all published, this nice diagram showing phosphate and nitrogen increasing concentrations, and you see that when you have low concentrations of both, you get these nice non-toxic strains and these cells with lower toxin quotas. But the more eutrophic your system gets both nitrogen and phosphorus, you're going to see a lot more elevated toxicity. And the reality is that we have been increasing the amount of reduced nitrogen exponentially. We have this global shift towards the use of urea and anhydrous ammonium as fertilizer. And urea is now potentially more than 50% of worldwide applications in terms of agricultural additions. And so, to me, this says that we're not regulating nitrogen, but we should be. It's important. It's important for toxicity, it's important for biomass, and it is important for community structure. If you want to push the system away from cyanobacteria and back towards diatoms, you got to decrease the amount of reduced nitrogen loading. So I'm going to talk about this a little bit more detail about the research that we're doing regarding this. And this is the nitrogen cycle. I realize it's complicated, it's got a lot of arrows. The best parts about working in the nitrogen cycle is we like to add a few arrows every couple of years just to mix things up. And here you can see that you start off with N2 gas, our difficult thing to fix here, but it can be fixed. And when that cell breaks down, it will release ammonium. Ammonium can be nitrified first to nitrite, and then nitrate, which under anoxic conditions can be denitrified back to N2 gas and theoretically effectively removed from the system. There's also an alternative removal pathway called Anomox. But also importantly, and I'm not going to talk about this today, but DNRA is a pathway that the more eutrophic the system gets, the more you shift from potential denitrification to Anomox. And when that happens, you are recycling your nitrogen instead of removing it from the system. But the two main things I'm going to talk about right now are these competition for ammonium pathways. So uptake and assimilation into organic nitrogen and then release back into the pool of ammonium, and removal of ammonium into oxidized nitrogen via nitrification. So my lab uses a paired molecular biogeochemical approach to try to break apart these crazy complicated relationships within the nitrogen cycle and try to understand how this insane amount of increased reactive nitrogen in the world is impacting nitrogen cycling. And so I'm going to focus on, like I said, this ammonium uptake and assimilation and regeneration or remineralization and nitrification. So nitrification was, until last year, like I said, we like to complicate things, a two-step process. First one being ammonia oxidation into nitrate oxidation, or nitrate, and then nitrate oxidized to nitrate. And we thought these were mutually exclusive sets of organisms that could do these two pathways, but now there are apparently some that can do something called common-mox, which is the whole thing in one straight shot. But it's important because as far as we know, and there's some evidence to the contrary, but as far as we know, it requires molecular oxygen as a substrate, so it can only be done under acidic conditions. And as a potential side pathway, it can produce N2O, so it's a greenhouse gas producer, which ramps up under low oxygen. But mostly this end product of removing things into nitrate means that it's then a critical link to both growth and nitrogen removal via denitrification. Ammonium is so much easier to use than pretty much everything else. Microcystis has a strong affinity for it. It's got a half saturation constant that can range from 0.5 to 37, which means not only can it be really good at taking it up under low concentrations, but there are strains that have a strong affinity but a higher Vmax, which means that they don't get saturated so easily. They can accommodate very high concentrations of ammonium in the water. And between their two kinds of ammonia oxidizers, archaea and bacteria, there's also a very wide range of ammonium uptake affinity and saturation. And so depending on who's there and who's able to fight over this ammonium, we're thinking that the potential competition between nitrifiers and Microcystis might tell us something about who's going to dominate in the system. So we've got some pretty basic research questions in the two lakes that we're looking at, many of which have not really been addressed very much, which are, what is the rate of nitrification? How fast does it go from ammonium to nitrate? And how quickly is that ammonium recycled if it's taken up by the phytoplankton community? Are archaea or bacteria responsible for ammonia oxidation, which we're working on, but I'm not going to talk to you a lot about it today. So far we're seeing more AOB than AOA, spoiler alert. And then to what extent can these ammonia oxidizers compete with phytoplankton and cyanobacteria? And to start with, I'm going to talk about some work that my Ph.D. student, Justina Hempel, is doing in Taihu in China. And these 4 stations here that I'm showing, 1, 3, 7, and 10, are going to be the main places that I'm looking. This used to be an inflow here, and now it's changed to an outflow based on some construction and engineering that the Chinese government has been doing to try to improve the lake. Taihu suffers from some very serious microcystis blooms. I took this picture on the left in August of 2013 when it was so hot. It was 140 degrees with the heat index. It was pretty insane. This middle picture that Hans took is right after the typhoon that happened also in 2013. And this is actually a core all the way on the right here. This is the potentially oxic surface sediment here and going down into very anoxic mud. And you can see the microcystis just sitting right on top of the core after we took it. I'm not going to talk any more about the sediment core work today. That's being done by my collaborator, Mark McCarthy. So we tried to measure nitrification using 2 different methods. And the first one is a pool dilution method where you add 15 nitrate and see how much new 14 nitrite is produced. The problem with this is that it relies on preferential uptake of 14 over 15, and so there might be some issues with it. But it's a nice way to look at nitrification because you are not adding potential substrate to the rate. So it shouldn't be at the potential rate, it shouldn't be increasing the rate, it should just be reflecting what's actually happening. And we do this by looking at both the ammonium concentration. We reduce the 15 and 14 nitrate all the way to ammonium, and we can measure that 15-14 ratio on the oxmims. And what you see is some very high rates. I want you to notice here that these are not in order of year, but in order of months. So you've got spring, March, June, August. And these are, again, total nitrification rates or nitrate production. And you're looking at nitrification rates in micromolar per hour, which is very fast. And so what you can see is that just it doesn't really matter all the stations and depths. The point is that as you're going from less bloom in the spring to lots of bloom in August, there's an overall decrease in rates. So our nitrifiers are probably not competing that well with the Microcystis. While their rates are extremely high, even in August for rates, they are definitely decreasing as you get into the bloom. And if we look at this a different way, where we add 15 ammonium tracer, and we see how much ends up as 15 nitrite and 15 nitrate. So this is now more of a potential rate because we've added substrate to it. We try to add not super high amounts, but there's so much ammonium in Taihu that we do have to add a significant portion at times. And these now are in nanomolar per day, so these rates are not as high when we look at it through the tracer addition, but they still, this would be 1 micromolar here. They're still pretty high. And what you can see is that not a lot of it shows up here in blue as ammonia oxidation. Most of it is showing up green here as nitrate oxidation. And this makes a lot of sense to us because ammonia oxidation is the rate limiting step. It happens more slowly, and once it gets converted from ammonium to nitrate, it gets nitrified from nitrate to nitrate almost instantaneously. Those bacteria are very fast at what they do. And there's a reason you don't see nitrite built up in the water column, right? You can measure ammonium, you can measure nitrate, but not so much nitrite. So pretty much as soon as the ammonia oxidizer is converted to nitrate, it gets converted to nitrate. And you see, we don't have August here because we didn't do it that first year, but now if you're looking at March, June, and July, you again see a trend of decrease as you're going from March into the bloom. And so when we're trying to figure out what's the actual competition here, how fast can the whole phytoplankton community, which does include microbes, take up ammonium, and then how fast does that get regenerated, remineralized, recycled, whatever word you want to use for it. And so what extent of the ammonium uptake is being supplied by recycling or turnover of ammonium. Yeah. And we do this by looking at ammonium uptake and regeneration. So we add 15 ammonium, and then we see how much new 14 ammonium gets produced. So again, it's a pool dilution method. And these graphs are a little bit complicated to look at, but what I want you to notice here is in blue, this is our uptake, and this is light versus dark, so you expect the light to be greater if you've got a lot of phytoplankton. And then the regeneration rate here is in purple, and again it's light versus dark. And then the nitrification rates down here are in green. What you see is that in June, at 2 of our stations, at the surface and in the deep depths, the nitrification rates are high, and when you compare them to the total uptake rates, nitrification can still account for a lot of the total ammonium uptake in June. And remember, these are now potential uptake rates, right, because we've added 15 ammonium, so we've increased the total ammonium concentration. And the regeneration rates are also pretty high, not everywhere, but they are often enough to supply at least a lot of the demand. And when you move into August of 2013 in the middle of the bloom, I'm unfortunately missing these uptake rates over here in August. Thank you, United Airlines, for breaking my cooler and destroying my samples. You can see that the total uptake rates now are much, much higher, and they are much higher than the nitrification here. So just compare the blue bars up here, the uptake in the light, to total nitrification rates down here. The nitrification rates haven't actually gone down that much, but what's happened is that the total uptake has just skyrocketed once you're in the bloom. And there's now more being recycled than even the nitrifiers can use. So there's a huge demand for ammonium within the bloom, and there's also a huge supply for ammonium within the bloom. This recycling and turnover can really sustain a lot of the ammonium demand of the whole community, but not all of it. So we see very high rates of nitrification in Taihu. We also see high concentrations of ammonium. Sometimes it does get down to about 1 micromolar, but we've also measured it as high as 137 micromolar. So I think in part these rates are so high because the in situ concentrations are so high. So we have these very high rates, but they definitely decrease during the bloom. And our ammonia oxidation rates are much lower in 2014 and 2015 than the total nitrification rates, but we think it's because they turn over too fast, and the nitrate oxidation rates, when you add to them, make the whole rate much higher. So I think that at some level, ammonia oxidizers can compete with phytoplankton and cyanobacteria for ammonium, but maybe not as well during the bloom, right? Because we see the rates being much higher during March than we do later. I also like to point out that this is farther south in China than we are currently here in Michigan, and so it's a lot warmer in March in Taihu than it is in Michigan, as in there's no ice cover. Alright, so now we're going to talk about Lake Erie. I don't think I need to tell anybody in this audience that we had a drinking water crisis in Toledo in 2014, or that we have massive annual blooms that are largely Microcystis in the western basin. But this is a fun video that Daniel took last year when he was out with Noah Gale. Sorry, I thought we got the volume turned down on that. Apparently not. Well, I won't show it to you then, but it was very gross, I promise, nice and scummy. So a lot of this is being driven by fertilizer additions. When we look at the Maumee Basin here, I know you can't read any of these things and it doesn't really matter. The point is that this color brown here shows you what's agricultural area in the Maumee River watershed. And this means that we are adding and increasing the amount of reduced ammonium going into Maumee. We have been for the last 15 years. And the killed-all nitrogen in 2010 was about a quarter of the total nitrogen load. We've been focusing on a few stations of the NOAA GLERL master stations, and this is all work that my Ph.D. student Daniel Hoffman has been doing. And the first year, 2015, we measured 6, 2, and 4. And then we discovered that 4, we were hoping to get a gradient away from the Maumee River. We discovered that 4 can be pretty strongly influenced by the Detroit River. And so this year we added 13, which should be a gradient away from both river mouths. So Daniel is looking at just the tracer additions. Overall, I think I'm more comfortable with them as a way of measuring rates. And so we add 15 ammonium and then measure it, how much it shows up as 15 nitrite and 15 nitrate. And if you look at Daniel's ammonia oxidation rates from 2015 and 2016, I want to point out that this scale only goes to 140 in 2015 and it goes to 1,000 in 2016. And this is just ammonia oxidation now. You can see the months progressing into the bloom. Probably I should have made August bright green, but you can see that before the bloom really hits is when you have the highest rates. And this is now blown up, so they're both on the scale of 0 to 50, just for comparison's sake of these lower numbers. And you can see that overall, even in August, and we've added April and May now in 2016, the rates are a lot higher in 2016 when the bloom was a lot lower. Now Daniel's working on the nitrate oxidation part to this. He has it for 2015, although it's not included here. And it does make the rates higher, but they still fit on this scale of up to 140. So I'm really curious to see what they're going to be for 2016. I think they will be much higher. But notice, again, this is in nanomoles per day and not micromolar per hour like we had for Taihu. But when you compare the total uptake and regeneration then, so this is, again, how much ammonium the entire community is potentially demanding. This is a potential demand because we've added the substrate, versus how much is actually being recycled and supplied. So the colors are different here on Daniel's graphs. You've got uptake here in the light and green, and then this is uptake in the dark, and then you got regeneration here in blue and regeneration in the dark. So got uptake, green and black, and regeneration in blue and gray. And this is from June of 2015 and Site 2, and we're back to being in micromolar per hour. So this is all 2015 now. We don't have 2016 yet. Daniel actually has the data, but we're still working it up. We were hoping to be able to show it to you, but unfortunately it's not going to happen. And in 2015, the bloom hadn't so much hit yet in June. And what you can see is that regeneration is not quite as high as potential uptake, but it is close to it. And maybe you're starting to break away a little bit more in the surface of the Maumee River Bay Station in 6, but overall you've got high uptake but high regeneration too. And when you get into July, when it's starting to get a little bit greener, you got a lot more uptake, and regeneration is trying, but it can't quite get into it. Notice too that the difference between dark and light in July versus June has really significantly changed. Not surprising, right, if we're going to more ammonia oxidizer use or bacterial use, something that isn't based on light. And then you've got a lot more phytoplankton growing in June. They need the light, and then the light they just don't do as much. Not particularly surprising. Now notice here that these scales go from up to 1.4 micromolar per hour. Now when we switch over into August, these rates are now up to 25 micromolar per hour in the height of the bloom here with all this surface scum. There's not a lot of difference between light and dark here because the incubation time we ended up using was only 15 minutes long because it took up the ammonium that quickly. But notice that even though we have very high regeneration rates now, I'm talking 5 micromolar per hour or so, who have even higher ammonium demand at this point. So they could, when you give it to them, take up a lot more ammonium that's being supplied. But even though we measure 0 concentration at this point in time, the regeneration rate is 5 micromolar per hour. There's a lot of ammonium being turned over here. And when the bloom backs off, you still see this nitrogen demand being so high here, these high potential uptake rates, and the regeneration has gotten much lower as the bloom biomass is dying off and sinking out or switching to somebody else. So we do see these very high ammonia oxidation rates, but they're not as high as Taihu. They are, however, pretty comparable to other areas, coastal areas, other lakes. And we see the nitrification rates are lower during the bloom year than they are during 2016, which was not as high of a bloom. But the difference between uptake and regeneration rates and ammonia oxidation rates is significant. The total community demand in Taihu is on the same order of magnitude as the nitrification rates, whereas in Erie they are per day versus per hour. So the per hour rate, the total ammonium demand is much higher than what those ammonia oxidizers are doing, and I think that some of that might have to do with the total ammonium concentrations that are available. I could be wrong, somebody tell me in this audience, but I don't think we ever see 137 micromolar ammonium in the western basin of Lake Erie. So we do also see this difference in uptake and regeneration rates within the total phytoplankton community throughout the bloom. See a much greater uptake and demand, but there's a much greater disparity between them as you progress in the bloom as well. And so before the bloom, we can have potentially uptake rates that are sort of balanced by what's being available, but the farther you get into the bloom, the more there is this increase in potential demand. And Wayne Gardner is spearheading an effort to look at the difference between this potential uptake and regeneration, and he's calling it the Community Biological Ammonium Demand, the CBAD, and we are, I think maybe have just submitted a paper about that, looking at it as a metric of how potentially nitrogen needy or ammonium limited the community is. So there have been a lot of people who have helped us with this work. We're very lucky to be collaborating both with several people at NOAA GLERL, including Tim and Tom. We're also working with Justin Chaffin at Ohio Stone Lab, and I've got another student who's been going out with him. And, she's working with my collaborator Mark McCarthy on sediment uptake and supply of nutrients, denitrification rates, etc. And they're looking at them at these same stations, so we'll be able to compare what's happening in the sediments to what's happening in the water column. But that's it. Thank you very much. I think that's true. I think policy and managers have been focusing on phosphorus. I do think, though, that some of the changes we've been taking in Ohio, and I assume you are in Michigan, are actually reducing nitrogen along with the phosphorus. We're talking about manure applications to fields, for example, or timing of fertilizer applications. That should impact both nitrogen and phosphorus. Oh, sorry. Yes, if managers were mostly focused on phosphorus, and I think that as long as that's the message that the scientific community is still putting out in terms of policy, that's what our managers are going to be hearing. Yes. Yes, please. Come on up.
I was involved in the development of the target phosphorus loads for Lake Erie, and we were definitely very aware of the nitrogen interactions and how that might potentially be affecting things. But we can't quantify that yet. We don't have models that will develop some sort of stressor response relationship so we can propose defensible targets.
Well, fortunately for you, I have developed a new relationship that hopefully will get published soon.
No, it was definitely on the radar, but the recognition that our phosphorus is actually quite good at that. So we took a lot of steam out of it, our challenge. But I think
I will say that Hans Perl has a pretty nice paper suggesting target nitrogen load reduction along with total phosphorus.
Is there any ammonium released by micrograzers in these bottle experiments?
Potentially, yes, we're looking. So he's asking about what are the role of micrograzers within the bottle experiments in terms of ammonium release. And we don't quantify who's releasing the ammonium or who's doing the turnover remineralization. We're just interested in the total supply. So yes, I assume there are some micrograzers in there. No, actually you get a much bigger, so you said during a bloom you get as much nitrogen supply or regeneration as you do uptake, and in fact it's very, very different. We got uptake, potential uptake demand essentially up to 25 micromolar per hour, and the recycling supply is only 5 micromolar per hour. The question is the bloom ends because they're running out of ammonium or nitrogen, and I would say that Steve Wilhelm's group looking at the genes that are turned on during the bloom show that the cells are starved for carbon, nitrogen, and phosphorus at the height of the bloom. So I wouldn't feel comfortable saying it's only nitrogen, no. Great, thank you.
First question from the webinar, what are your suggestions to control nitrogen loads into Lake Erie?
Well, I think that some of the steps that we're taking in Ohio in terms of timing and not applying manure or other fertilizer on top of snow or right before it's going to rain, those are very important, and they apply to both nitrogen and phosphorus. I also think that judicial targeted fertilizer additions are going to be the way of the future. This is now my own personal opinion, but Mike Beeman and Pam Matson have done some interesting work in Mexico, and the farmers there have these little sensors they put on the leaf, and if it's not green enough they add fertilizer. It seems to me that that would be a pretty straightforward way to go in terms of combating the more is better mentality. Nobody wants to waste money, least of all farmers. Thank you very much.
Okay, thank you everyone, and thank you everyone who participated online. For those of you who are online, we appreciate it, and for those of you who are in the room, if you have not signed in to the seminar, please put your name on the yellow pad in the back. Also, if you're interested in going to lunch with the speaker, please feel free to meet us in about 15 minutes just at the front of GLERL. Lunch is on your own, unfortunately, but we are happy to accommodate whoever would like to come. So with that, that'll conclude today's seminar speaker, and thank you all for participating.
Muted.